We have Donald Hendrix, and he's going to talk about hydroxyl radical generation of lunar dust analogs in biologically relevant human respiratory system fluids.
Give me a second. Thanks, Douglas.
Yes.
Hold on, give me a second. Okay, perfect. Okay, thanks. Okay, so the point of my work is to understand the health effects of lunar dust. And so how I do that is I assess the reactivity or the OH radical generation of different minerals and lunar simulants and try to understand the potential inhalation health risks. So as John said, reactivity can mean many different things. It can mean generation of peroxide, generation of superoxide. I'm primarily focused on OH radical generation. And the reason I focus on that is because OH radical has been known to be cancerous to lung tissues.
Hey, Donald, real quick, this is Mike. We're seeing your presenter view at the moment. If you want to either swap displays or hit the mirror mode.
So actually, let me, oh boy. Let me do, let's see. Does that work?
Yes, thank you so much.
Oh, awesome. Yeah, thanks. Okay, so I'm part of the RISE 2.0 team, and within that team, I'm part of the Theme 3 group. And so what the Theme 3 group does is we try to understand the health effects of lunar dust through a variety of methods. First, we're trying to develop the next generation of lunar simulants, and by that, trying to make current lunar simulants more lunar-like, like by adding metallic iron, which I'll talk about in a bit later. And so, what we also do is we assess the reactivity of these simulants, which is what I'm going to talk about today. In the next talk, Jamie's going to talk about evaluating the toxicity of these lunar simulants. And so far what we've done is we've obtained 3 separate lunar stimulants, the traditional JSC1A. We've obtained LHS1 from the Exolith lab, as well as LMS1 from the Exolith lab. And since these simulants by themselves do not have metallic iron, we try to add some metallic iron. So basically how I do that is I take like a little silica boat and I add a little bit of the simulant sample, and that silica boat goes into the glass tube. And this glass tube gets flushed with hydrogen. Can you guys see my mouse?
Yes.
Awesome, yeah, okay. Okay, so yeah, so this tube gets flushed with hydrogen gas, and then this tube gets placed into the furnace. And that's heated to 900 C for 15 minutes. And then we remove our samples once they're cooled down. And we notice that they are visibly darker. And under SCM, we see all these little blebs on the surfaces of the grains. And these grains are metallic iron. There was a paper by Allen et al. 1994, which they used a similar method that we used, and they were able to determine through SCM and Mossbauer spectroscopy that these little blebs on the surfaces of these grains are metallic iron. And this is an SCM image of JSC1A grains. And in addition to that, what we've done is we've assessed the reactivity or the OH radical generation in 3 separate fluids. We did first in DI water, then we did it in simulated lung fluid, and then in artificial lysosomal fluid. And so simulated lung fluid simulates like the fluids in your air passageways, and artificial lysosomal fluid replicates the fluids in your macrophages. So I have here an SCM image of some macrophages. And so we're trying to simulate sort of what's inside of those and how reactivity would be affected once those macrophages engulf any inhaled particulate matter in the human lungs. So here's just the list of what's in both the simulated lung fluid and artificial lysosomal fluid in grams per liter. I highlighted in green the amino acids, and in red, DPPC is a surfactant that is commonly found in lung fluid. And so how we measure OH radical is through a technique called electron paramagnetic resonance, or EPR spectroscopy. And so basically what we do is we take about 100 meters squared per liter of sample, which that's based on the surface area of the dust simulants. And then we incubate that in 0.5 milliliter of an organic compound known as DMPO. So this is the organic molecule, a DMPO, and that will react with any hydroxyl radical generated in solution by any of the 3 simulants. And that will form this stable organic radical compound, which can then be measured very easily using the EPR. because we can't measure OH radical directly because OH radical is very reactive, so we have to sort of trap it. And that's what the DMPO does. So here are the results of reactivity in DI water. So R denotes the heat treated and reduced samples, and we highlighted the results of the reduced samples in red. And so as you can see, heat treatment and reduction of these samples leads to a large increase in the OH radical generation compared to the not reduced samples in the gray. And so this is very likely due to the presence of metallic iron, and kind of shows what how drastic of an effect the presence of metallic iron alone can have. And there was the other paper that John also talked about, Wallace et al. 2010, where they tested the reactivity of various Apollo samples and compared that to fine grain JSC1A, and they were able to see that there was a huge increase in the reactivity compared to the fine grain JSC1A. And they were also able to strongly correlate the reactivity with the maturity, or pretty much the amount of metallic iron that was present. So the advantage of this is that we can make these samples in the lab versus having to actually get pristine lunar regolith. And so this is where it gets kind of interesting. This is not an incomplete graph. So this actually shows the results in ALF and SLF, and we actually were not able to get a measurable signal in either of these fluids. But that doesn't necessarily mean that we're not making OH radical. It could also be that we're not making OH radical. But in the next slide, I'm going to go over some bullet points on what may possibly be happening. And so first, we saw in DI water that heat treatment and reduction leads to a sharp increase in the reactivity. And that's very likely due to the presence of metallic iron. So there was no measurable signal in the simulated lung fluid or the artificial lysosomal fluid. So the first point I have here is that it may be possible that iron on the surface of these grains are being chelated, or sort of being bound to any of the electrolytes present in both of these simulant fluids. And so if that iron is being bound, it's not able to participate in any type of chemistry that leads to the generation of OH radical. In the opposite case, what could be happening, the amino acids in both of these biological fluids, especially in the SLF, since SLF has more amino acids, is reacting with the OH radical at a rate much faster than that DMPO molecule can trap the OH radical for measurement. So if that's happening, there's no way that we can measure it using EPR. And on top of that, the sort of the negative health implication of the OH radical reacting with the amino acid is that since your proteins are made up of amino acids, if you inhale any lunar dust that immediately starts generating massive amounts of OH radical, that's going to interact with all the amino acids that make up your proteins. So then your proteins may start to sort of unfold and denature and lead to a whole bunch of other, bad stuff. And as for the artificial lysosomal fluid, it includes compounds like sodium tartrate, very high levels of sodium hydroxide and citric acid. And so it's possible that some of these reactive sites on the surfaces of these dust grains may sort of, in essence, be washed away or dissolved before it has a chance to make OH radical. And so to conclude, heat treatment and reduction leads to massive increases of OH radical in DI water, at least, and that's very likely due to the presence of metallic iron. So we were not able to measure OH radical in simulated lung fluid or artificial lysosomal fluid. However, that does not mean that we're not making OH radical. It could be that we're not, but in all honesty, it could be, a mix of both. It could be there's some components in the biofluids that are preventing OH radical from being made. And there's other components like the amino acids in the biofluids that are very rapidly reacting with the OH radical. And therefore we're not able to measure it because the DMPO reaction with the OH radical is much slower than that of the OH radical and the amino acid. And currently we are awaiting Mossbauer data for reduced simulants, and that will give us a much better idea of how much metallic iron we have in our samples compared to actual lunar samples. And with that, I'll take any questions. Thank you.
Thank you, Donald.
Oh, wait, should I reshare? Sorry, I kind of.
Does anybody have any question, Donald? I would have a quick question. In terms of comparing maybe the effects of in situ lunar regulator on those biofluids, do you have an idea, or how could you like perform experiments on those, not losing the toxicity or chemical activity of the dust specimen?
Like you're talking about, you said in situ, like on the actual lunar surface?
Yes. Would there be any plans to do that?
There's not any plans that I'm personally aware of, but that would honestly be the best option, because it's really difficult to replicate actual lunar dust. And I think there was a whole session sort of on that. And so, having, an instrument up on the surface would actually be superior, because we could sort of get, what, what effect, do these actual lunar dust samples have, on the human body. I think there was an instrument called Luna Chem, and I think John, actually, you were looking at that, right?
Yeah, we were working on developing an in situ measurement device. Yep.
Yeah, no, I mean, in situ measurements would be ideal, because also a lot of these Apollo samples that are being used, were not necessarily stored in the best environments. So that's something also that has to be kept in mind as well. And it's very difficult to replicate agglutinates and other things. So yeah, that would be very ideal.
All right. Yeah. Thank you.